Welcome back to the deep dive. Today, we are opening, well, probably the most complex instruction manual ever written, the human genome. Think about it. 46 chromosomes, all perfectly paired, meticulously folded up inside our cells, holding the blueprint for, well, everything about us. It's kind of an architectural wonder, really.
It absolutely is.
But, that architecture isn't always perfect. We often talk about the numerical errors having an extra chromosome 21, for instance, leading to Down syndrome. Today, though, we're going deeper. We're looking at the actual construction flaws. What happens when the pages of that manual are ripped out or maybe rearranged, parts lost, entire sections flipped upside down? We're talking about the architectural damage, the kind that changes the sequence and organization, even if the total number of pages, the chromosome count, stays the same.
And that's the critical distinction, yeah. We are taking a deep dive specifically into structural abnormalities of chromosomes. These physical changes, the breaks, the fusions that maybe didn't repair correctly, the exchanges of material, they're often much harder to spot than just counting chromosomes.
Right.
But their impact,
Yeah.
just as profound. And these subtle breaks, these rearrangements, they aren't just some, academic curiosity. They are often the root cause of things like high rates of spontaneous abortions or unique, sometimes really complex developmental syndromes. And they can also explain significant inherited health issues that might suddenly pop up in a family with seemingly no prior history of the condition. It really highlights how fundamentally fragile our genetic material can be.
Okay, so let's unpack this. Our mission today is to really get a handle on the precise language, it's almost like surgical language, isn't it, that cytogeneticists use to describe these faults.
It is, yeah.
And then dive into the mechanics, the nuts and bolts of how these structural changes actually happen at the molecular level, and ultimately connect those microscopic faults to the very real-world consequences for the individual, and sometimes, for their children, too. We're moving beyond just the count and really focusing on the content and its arrangement.
Exactly, content and structure. So to understand the faults, the structural problems, we first have to understand the map itself.
Right.
And the technique used to create that map, the foundational tool, really, of cytogenetics, is karyotyping.
Karyotyping. And the process itself, it always sounds wonderfully old school, but also incredibly specific. You basically have to catch the chromosomes at just the right moment, right? When they're all condensed and visible.
That's exactly it.
But that's a pretty quick window, isn't it?
It is. It's a fleeting moment in the cell cycle, which is why we need some chemical help. The standard procedure involves dividing cells, usually white blood cells, lymphocytes,
Okay.
and arresting their division using mitotic spindle inhibitors. The classic one is coulsomid.
Coulsomid, what does that do exactly?
So coulsomid basically messes with the machinery, the spindle fibers, that pull chromosomes apart during cell division. It prevents the cell from completing mitosis. Specifically, it freezes them right in metaphase. That's the sweet spot. That's the only time they're condensed enough, tightly packed enough to actually see clearly with a standard microscope.
You mentioned arresting the cells. It almost sounds like it's just by chance we can see them at all. What happens if you miss that metaphase window, like if you're too early or too late?
Well, if you miss it, if you're too early, they're still elongating. If you're too late, they've already started pulling apart or decondensing. Outside mitosis, the DNA is just this big tangled mass we call chromatin. You can't resolve individual chromosomes or count them accurately at all. So coulsomid ensures they condense fully, line up nicely at the metaphase plate, but stops them before they try to pull apart into two new cells during anaphase. Once we harvest these arrested cells, we spread them on a slide, stain them, and then under the microscope, we can clearly see the individual chromosomes. Each one usually looks like an X shape, well, not always an X, but composed of two identical sister chromatids joined at that pinched-in point, the centromere.
Okay, so you've got the picture. Then the real work begins, building the karyotype itself. This is where you bring order to that chaos, right?
Exactly. The process involves taking a high-resolution photograph, or more commonly now, a digital image. Then you digitally cut out the individual chromosomes.
The little puzzle pieces.
Kind of, yeah. And you arrange them very systematically. The 22 pairs of autosomes, those are the non-sex chromosomes, are arranged strictly by length. From the biggest, chromosome 1, all the way down to the smallest, chromosome 22. And then right at the end, you place the sex chromosomes, either XX for females or XY for males. This standardized arrangement, this map, allows for a quick visual comparison against a normal reference. You can immediately see if a whole chromosome is missing or extra.
But as we said, we're going beyond just counting today. Just looking at the overall size and shape isn't nearly enough to spot something subtle, like a small rearrangement or a tiny piece missing, right?
Absolutely not.
That's where chromosome banding comes in, I guess, providing the resolution to see the internal structure.
Precisely. You need a way to tell chromosomes of similar size apart. Like, how do you reliably distinguish a chromosome 10 from a chromosome 11? They look pretty similar in overall size.
Good point.
So the most common method, the workhorse, is G-banding. It uses a stain called GIMSA stain.
GIMSA. Oh.
And this stain interacts differently with different regions of the chromosome based on the underlying DNA composition, the proteins.
Yeah.
It creates these highly reliable, alternating patterns of light and dark bands.
Ah, like stripes.
Exactly, like stripes. Unique to each chromosome pair, it essentially acts as a microscopic barcode or a fingerprint for each chromosome.
Okay, so that's how we identify them. But what about the detail? How much can you actually see with standard G banding?
With standard G banding, which is typically done on those metaphase cells we talked about, you can usually resolve somewhere between 400 to maybe 800 distinct bands across the entire haploid set. That's one copy of all the chromosomes.
400 to 800 bands.
Yeah. And for identifying, whole chromosome swaps or large numerical errors like trisomy 21, 400 bands is often perfectly sufficient.
Okay, but here's where it gets really important for what we're discussing today, right? Because 800 bands might sound like a lot, but sometimes these structural flaws, they're tiny. Maybe a deletion of only a few genes. Could that fall between those standard bands?
That is the major limitation, absolutely. And that's precisely why high-resolution banding techniques were developed.
Ah, okay, so how do you get high resolution?
The trick is actually to catch the cells earlier in the cell cycle, specifically in prophase or maybe prometaphase. This is before they've fully condensed into that tight, compact metaphase structure.
So they're longer and more stretched out.
Exactly. You get markedly elongated, longer chromosomes. Think of it like stretching out a coiled spring. You can see the individual coils much better.
Okay, that makes sense.
This elongation allows cytogeneticists to recognize significantly more bands, potentially up to 1,500 bands per karyotype.
Wow, nearly double.
Yeah. And this high resolution is absolutely essential for spotting those minute structural flaws, the very small interstitial deletions or maybe a balanced inversion that might have major clinical impacts but would be completely invisible, completely cryptic on a standard 400-band spread.
It's like the difference between reading just the chapter titles versus reading every single sentence in the book.
That's a great analogy, yeah.
So once we have this beautiful high-resolution map, we need a consistent way to talk about what we find, a language that works in labs all over the world. This is the cytogenetic nomenclature, right? It's almost like giving every single gene location a global GPS coordinate.
It is. It's a crucial international shorthand system. And the description follows a strict, universally recognized order.
Okay, what's the order?
First, you state the total number of chromosomes found. Second, you list the sex chromosome complement XX or XY. And finally, you describe the specific abnormality, if any.
Okay.
So for instance, a male with the common numerical form of Down syndrome, the one with an extra chromosome 21, would be written as 47XY plus 21.
47 total chromosomes, XY male, plus an extra 21. Simple, unambiguous.
Exactly. But for structural changes, it gets a bit more detailed.
Right, because you need to describe where the change happened, and this involves demystifying the chromosome arms. I know the basics, but let's quickly review why we call them P and Q.
It's surprisingly simple, actually, but crucial for precision. Each chromosome has that pinched-in part, the centromere. The arm above the centromere, which is typically the shorter arm, is designated P.
P.
And that P comes from the French word for small, petit.
Ah, petit. Okay, P for petit, short arm.
Exactly. And the arm below the centromere, usually the longer arm, is designated Q.
And Q stands for?
Nothing special, actually. It's just the next letter in the alphabet after P.
Oh, okay. That's less romantic than petit. So P is short, Q is long.
That's the convention. And from there, the system details the exact address, getting incredibly specific. Each arm, P and Q, is divided into regions. These are numbered 1, 2, 3, and so on, moving outward from the centromere.
Okay, regions numbered from the centromere out.
Then each region is subdivided into bands, which are also numbered. And those bands can be further broken down into subbands, again numbered.
Wow, okay. Before we hit the example, let's just lock down this GPS system. Why count outward from the centromere? Is there a functional reason, like region 1 near the centromere is different from a band way out near the end, the telomere?
Well, the numbering system itself is mainly for mapping consistency, just to standardize the location so everyone knows exactly what spot we're talking about. Functionally, yes, the location does matter. Regions near the centromere, the proximal regions, often have different types of chromatin, different gene densities than regions near the telomeres, the distal ends.
Okay.
But for describing a structural break, the absolute key is just knowing the precise breakpoint coordinates. We need to know if it's, say, XP21.2 versus XQ21.2. Short arm versus long arm makes a huge difference.
Okay, so let's use the sources example to see how precise this gets. XP21.2, break that down for us.
Right, so XP21.2 translates very precisely to the X chromosome,
Okay.
the short arm.
Right, petit.
Located in region 2.
Moving out from the centromere.
Within that region, it's band 1. And specifically, within that band, it's subband 2.
XP2 1.2, wow.
That level of precision is absolutely necessary. Because when we talk about a tiny deletion causing a major syndrome, like the 22q11.2 deletion we'll discuss later, everyone needs to agree exactly on what genetic real estate is missing, right down to the subband level.
Makes sense. Without this uniform language, clinical geneticists, researchers, they could never reliably communicate findings, track familial patterns, or even compare results across different studies.
Exactly. It's the common tongue of cytogenetics.
Okay, so we know how to map them, how to describe them. Now let's talk about how these faults physically happen. The source material makes it pretty clear that most structural changes start with the same basic event, a chromosome actually breaking.
Exactly. Breakage is usually the initiating trigger, and then following that break, one of several things can happen. Either the broken piece, the genetic material, is lost entirely, or it gets rearranged, maybe reattached, but in an incorrect way. We also need to remember, and this is important context, that most errors involving a significant loss of genetic material are just too severe. They're incompatible with life and usually result in an early spontaneous abortion.
Right. So what we see clinically are the ones that somehow slip through or were less severe.
Precisely. And before we dive into the specific types of rearrangements, let's just underline that detection limit again with standard banding. We said it's blunt. It requires a pretty large loss, something like 2 to 4 million base pairs to even be visible. Why do we keep emphasizing that large size?
It sounds huge.
Because the sheer amount of genetic information packed into, say, 2 million base pairs is massive. You could have dozens, maybe hundreds of genes in that stretch.
Wow.
So missing even a tiny fraction of that, maybe just 50,000 base pairs, 50 kilobases, could mean losing a critical gene or a crucial regulatory element. And that kind of loss would be completely missed by standard G-banding.
Which is why we needed the newer molecular tools like ESH and sequencing to find those smaller microdeletions that fall below that 400-band resolution limit.
Exactly. That's where the field had to go.
Okay, so let's start with the simplest type of structural fault, deletion, just losing a piece.
Right. Deletion is defined simply as the loss of a portion of a chromosome. And we generally see two main mechanical types, depending on where the breaks happen.
Okay.
The first is called an interstitial deletion. This requires two breaks happening within a single chromosome arm.
Two breaks in the same arm.
Yes. The piece of the chromosome between those two breaks just lost. It floats away, gets degraded, and then the broken ends on either side of the gap fuse back together.
Ah, so it's like cutting a piece out of the middle of a rope and then tying the remaining ends together. The rope is now shorter.
That's a perfect analogy. It shortens the chromosome arm.
So if that interstitial piece is lost, what actually dictates whether the remaining ends successfully fuse? Does that always happen cleanly? Or is this fusion process itself kind of error-prone?
It's actually quite error-prone. The cell has repair mechanisms, primarily one called non-homologous end joining, or NHEJ. It's designed to be incredibly fast at sticking broken DNA ends back together, which is good for preventing total chromosome disintegration.
Right.
But NHEJ prioritizes speed over accuracy. It's kind of sloppy. Sometimes it might nibble away a few extra base pairs before joining, or join ends that were originally next to each other. So while it often does fuse the ends after an interstitial deletion, the repair itself isn't always perfect.
Got it. And the notation reflects this precision.
Yes. So an example notation might be 46XY-del16, P11.2, P13.1.
Okay, let's decode that. 46 chromosomes, XY male, del for deletion.
Right, and the deletion is on chromosome 16. Specifically, the parentheses tell you the break points on the short arm. The first break was at band T11.2, and the second break was further out at P13.1.
So everything between P11.2 and P13.1 on that arm is gone.
Precisely. That segment is lost.
Okay, what's the second type of deletion?
The second type is a terminal deletion. This results from just a single break happening somewhere along a chromosome arm.
One break.
One break. The fragment that breaks off the end piece, the terminal fragment, critically lacks a centromere.
Ah, and the centromere is needed for?
For attaching to the spindle fibers during cell division. Without a centromere, That broken-off fragment can't be properly segregated into daughter cells, so it's inevitably lost during the next cell division.
Okay, so the end piece is gone. What about the main part of the chromosome that's left? It now has a raw broken end. Isn't that dangerous?
It would be, yes. Raw DNA ends are seen as damaged by the cell and can trigger repair mechanisms or even cell death. But, crucially, the remaining retained chromosome usually acquires protective sequences, called telomeres, onto that newly broken end.
Oh, it heals itself by adding a telomere cap.
In essence, yes. Specialized mechanisms add telomeric sequences, which protects the chromosome from being further degraded or, importantly, from fusing inappropriately with other chromosomes.
Fascinating. Okay, so moving from simple loss to something that sounds more visually dramatic, the ring chromosome. You describe this as an extreme coping mechanism for a chromosome broken at both ends.
A ring chromosome is exactly that. It's a special and quite dramatic form of deletion. It happens when breaks occur at both ends of a single chromosome. One break on the short arm, P, and one break on the long arm, Q.
Both ends snap off.
Yes. And then the damaged, sticky ends of the main chromosome body, where the breaks occurred, fuse together. They circularize, forming a ring structure, like a tiny genetic rubber band.
Wow. And obviously, if significant genetic material was lost from those initial terminal breaks, you'd expect phenotypic abnormalities, just like with other deletions.
Correct. The amount of material lost from the ends before fusion is key to the clinical outcome.
But beyond just the loss of material, why is a ring structure itself particularly problematic for the cell? Does it cause issues just by being a ring?
Yes, they are notoriously unstable biologically. Ring chromosomes just don't behave normally during mitosis or meiosis. Imagine the cell trying to replicate that ring and then pull the two resulting sister rings apart during cell division.
It sounds like a recipe for tangles and breaks.
It is. They often get tangled, break, or fail to segregate properly into the daughter cells. This can lead to cells dying off or to mosaicism, where some cells in the body have the ring and others have lost it or have rearranged versions.
Yeah.
They're fundamentally structurally disadvantaged when it comes to cell division.
And the notation for this is simple.
Very simple. For example, 46XYR14, that just indicates a male karyotype with a ring formed from chromosome 14.
Okay, next up, the inversion, the flip. You mentioned this is the ultimate example of a structural change that might seem harmless for the person carrying it.
Often, yes. An inversion also involves two breaks within a single chromosome. But critically, instead of the segment between the breaks being lost, it gets reincorporated, but only after being flipped 180 degrees.
So it's put back in, but upside down.
Exactly. If you imagine the chromosome's band pattern as that barcode, a section of that barcode is now running in reverse order.
And we need to contrast the two types here. Paracentric versus pericentric. What's the difference?
The difference hinges on whether the centromere is involved in the flipped segment. A paracentric inversion involves two breaks that occur on the same arm of the chromosome, either both on the P arm or both on the Q arm. Crucially, the centromere is not included within the inverted segment.
Okay, parallel to the centromere maybe. Doesn't include it.
You could think of it that way. Now, a pericentric inversion, conversely, involves breaks on opposite sides of the centromere, one break on the P arm and one break on the Q arm. So the segment that flips includes the centromere itself.
Around the centromere.
Exactly. And pericentric inversions are often easier to detect visually in a karyotype because flipping a segment that includes the centromere can sometimes change the relative length of the P and Q arms quite noticeably. The arm ratio might look different.
Okay, so if I have a page flipped upside down in my instruction manual, but all the words are still there, just in a weird order on that page, maybe the overall machine still functions. The source points out these are often fully compatible with normal development because no genetic material is typically lost or gained. The person is phenotypically normal.
That's exactly right. For the individual carrier, the gene dosage is correct, all the genes are present, they're typically perfectly healthy. This makes the inversion potentially cryptic hidden.
But you also called it a genetic time bomb. If the carrier is normal, how do we even find these inversions? When does the bomb go off?
That is the critical clinical challenge. We usually only detect these balanced inversions when the carrier runs into problems with reproduction.
Fertility issues.
Yes, often during investigations for recurrent spontaneous abortions, or sometimes when they have a child who is born with developmental problems because they inherited an unbalanced version of the parent's inversion.
Unbalanced? How does that happen?
It happens during meiosis, the process of making sperm or eggs. During meiosis, homologous chromosomes, the matching pairs, need to line up perfectly, band for band, so they can exchange genetic material in a process called crossing over.
Okay.
But if one chromosome has an inverted segment, it can't line up properly with its normal partner in that region. To try to make it work, the chromosomes contort themselves into an elaborate inversion loop structure.
A loop. Okay.
And if crossing over, that exchange of material happens within that loop. Well, that's where the trouble starts. It generates gametes, sperm, or eggs that are highly unbalanced. They end up with duplications of some segments and deletions of others.
So the resulting embryo gets either too much or too little genetic information.
Precisely, which usually leads to non-viable pregnancies, spontaneous abortions, or offspring with significant congenital abnormalities. The seemingly harmless inversion in the parent becomes a major risk factor for their children.
Wow. Okay, the next mechanism is isochromosome formation. This sounds like a really neat but disastrous way for the cell to lose one whole arm while simultaneously duplicating the other arm.
It is a very specific type of error, usually happening during cell division, either mitosis or meiosis. Isochromosome formation occurs when the centromere divides incorrectly. Instead of dividing longitudinally, separating the two sister chromatids,
Like it normally should.
it divides transversely, horizontally, across the centromere.
Sideways.
Yeah, sideways. The result is that one entire arm of the chromosome, say the P arm, is lost completely, and the other arm, the Q arm in this case, gets duplicated. So you end up with a new abnormal chromosome that consists of two identical long arms, two Q arms, and no short arm at all. Or vice versa, two short arms and no long arm.
Whoa. So you get a complete loss of all the genes on one arm, that's a monosomy for that arm, and at the same time you get a triple dose of all the genes on the other arm, a trisomy for that arm. That's a huge genetic imbalance from one small error at the centromere.
That's the exact genetic result. The two arms of the isochromosome are mirror images, genetically identical. It creates this simultaneous massive loss and massive gain of genetic material.
Is there a common example of this?
Yes. The most common isochromosome seen in live births involves the X chromosome. It's designated IXQ10. This means it's an isochromosome of the X chromosome formed from two copies of the long arm Q with the breakpoint effectively at the centroband Q10.
IQ10.
So an individual with this has monosomy for all the genes on the X chromosome's short arm, the P arm is lost, and trisomy for all the genes on the long arm. They have two Q arms plus the Q arm on their other normal X if female. This has severe clinical implications, particularly related to Turner syndrome, which we'll get into later.
Okay. Finally, let's tackle translocations, the swap, the exchange of material between different non-homologous chromosomes. Two critical patterns here, right?
Right. The first is the balanced reciprocal translocation.
Reciprocal meaning back and forth.
Exactly. This mechanism usually involves single breaks occurring in each of two different non-homologous chromosomes. Let's say a break on chromosome 2 and a break on chromosome 5.
Okay, two different chromosomes break.
Yes. And then they essentially exchange the broken pieces. The fragment that broke off chromosome 2 gets attached to chromosome 5, and the fragment that broke off chromosome 5 gets attached to chromosome 2.
A clean swap.
Usually, yes.
Yeah.
And again, the clinical significance for the carrier is that they're typically phenotypically normal. Why? Because little or no genetic material is actually lost or gained in the exchange. The overall genetic content is still balanced, it's just located in the wrong chromosomal spot.
Balanced but rearranged. Okay, I sense another but coming.
You sense correctly. The hidden risk is, once again, immense, and it manifests during reproduction, during meiosis.
The pairing problem again.
The pairing problem, exactly. During meiosis, these two translocated chromosomes, the rearranged 2 and the rearranged 5, and their two normal partners, the normal 2 and the normal 5, all have to try and pair up together. To do this, they're forced into a complex, cross-shaped structure called a quadrivalent.
A quadrivalent.
Okay. Sounds complicated. It is. And when the cell tries to segregate these four chromosomes into gametes, it can do so in several different ways. Many of these segregation patterns result in gametes that are unbalanced. They might get, say, a normal 2 and a translocated 5, leading to duplications and deletions.
So the carrier is highly prone to producing abnormal sperm or eggs.
Very prone, leading again to a high risk of spontaneous abortion or having children with congenital malformations due to the unbalanced karyotype. The notation for this, like 46XXT25Q31P14, tells you it's a female XX with a translocation between chromosome 2 and chromosome 5 and specifies the exact breakpoints on the long arm of 2Q31 and the short arm of 5P14.
Okay, now the second, and maybe clinically the most famous type of translocation, the Robertsonian translocation, also called centric fusion, famous for its role in some cases of Down syndrome.
Yes, Robertsonian translocations are a special case. They occur only between acrocentric chromosomes.
Acrocentric, remind us what those are.
Acrocentric chromosomes are the ones where the centromere is located very near one end, resulting in one very long arm, the Q arm, and a very, very short arm, the P arm, that often just looks like little knobs or satellites. In humans, these are chromosomes 13, 14, 15, 21, and 22.
Okay, 13, 14, 15, 21, 22.
Got it. So in a Robertsonian translocation, the breaks happen at or very close to the centromeres in two of these acrocentric chromosomes. It's a chromosome 14 and chromosome 21.
Okay.
The two long arms, 14p and 21q, fuse together, forming one large, essentially complete chromosome, and the two tiny short arms, 14p and 21p, also fuse together, forming an extremely small fragment.
And what happens to that tiny fragment?
That tiny fragment, containing only the short arms, is usually lost entirely during subsequent cell divisions. It just doesn't have enough genetic clout to be maintained.
It just disappears. But wait, isn't losing a whole chromosome fragment bad? Why is the carrier of a Robertsonian translocation usually phenotypically normal?
That's the crucial point. It turns out that the short arms of all 5 acrocentric chromosomes carry very similar, highly redundant genetic information. Mostly, they contain multiple copies of genes that code for ribosomal RNA, rRNA.
Ribosomal RNA, needed for making proteins.
Yes, essential for building ribosomes. But we have so many copies of these RNA genes spread across all the acrocentric chromosomes that losing the ones on that tiny fused fragment is functionally negligible. The person still has plenty of RNA genes on their remaining acrocentric chromosomes, so the loss is compatible with the normal phenotype.
Fascinating. So they effectively have 45 chromosomes, one of which is this large fused one, but they have the right dosage of essential genes.
Exactly. They are balanced carriers. And this translocation is surprisingly common, detected in about 1 in 1,000 apparently normal individuals. But as you mentioned, its key clinical significance lies in its role in causing a familial form of Down syndrome, which isn't related to maternal age. So we've seen the mechanics. It's clear that the clinically recognizable disorders we actually encounter are really just the tip of the iceberg. They represent that small fraction of errors, maybe related to that 7.5% of conceptions figure, that are actually compatible with survival past early pregnancy. Yeah. Structural integrity is obviously paramount.
Right. So let's see these faults in action now. Let's connect the mechanisms to some major clinical examples.
Good idea.
We can start with one of the most common structural disorders mentioned, chromosome 22q11.2 deletion syndrome. The source notes this occurs in as many as 1 in 4,000 births. That's actually quite frequent for a specific genetic syndrome.
It is surprisingly common, yes. And it's a classic, really illustrative example of the power of a relatively small deletion, what we call a microdeletion.
Microdeletion.
It's caused by the loss of a small segment of DNA, specifically from band Q11.2 on the long arm of chromosome 22. What makes this condition so compelling from a historical perspective is that before we had the molecular tools, like Messier, to actually see this specific deletion, doctors were diagnosing what they thought were 2 entirely separate syndromes based on the clinical features.
Right. Those are DiGeorge syndrome and velocardiofacial syndrome, or VCFS.
Exactly. DiGeorge syndrome was typically characterized by problems with the immune system, specifically the thymus gland not developing properly, thymic hypoplasia, leading to a T-cell immunodeficiency. And also endocrine problems, like the parathyroid glands being underdeveloped, parathyroid hypoplasia, causing low calcium levels, hypocalcemia.
Okay, immune and calcium problems.
Then velocardiofacial syndrome, VCFS, emphasized a different set of features: heart defects, cardiovascular anomalies, problems with the palate, like cleft palate, Vila refers to the soft palate, and characteristic facial features, facial dysmorphism.
They sound quite different. Heart and face versus immune and calcium.
They do sound different. And for years, they were treated as distinct entities. But then, with the advent of higher resolution cytogenetics, and FIS, researchers realized that the vast majority of patients diagnosed with either DiGeorge or VCFS actually shared the exact same underlying genetic cause. That small deletion on chromosome 22q11.2.
Wow. So for decades, doctors were essentially looking at different facets of the same underlying architectural flaw. Was there a key gene discovery that really unified the picture? You mentioned TBX1 earlier.
That was a huge part of it, yes. The deleted region itself isn't tiny at the molecular level. It's about 1.5 million base pairs, 1.5 megabases, and contains somewhere between the 30 to 40 different genes.
Okay, so losing 30, 40 genes could explain the variable symptoms.
Exactly. The clinical heterogeneity, why some patients have more severe heart problems while others have more pronounced immune issues, likely reflects which specific genes within that deleted segment are most critical for certain developmental pathways, maybe influenced by other genetic factors or even environmental exposures.
But TBX1 is central.
TBX1 seems to be the major player. It's a type of gene called a T-box transcription factor.
Transcription factor, meaning it controls other genes.
Precisely. It acts like a master switch, turning other genes on or off during development.
Mhm.
And TBX1 is known to be critically expressed in the embryonic structures called the pharyngeal arches, specifically the third and fourth arches.
And those arches develop into.
Into many of the structures affected in this syndrome. The heart, the major blood vessels coming off the heart, the face, the palate, the thymus, and the parathyroid glands.
Ah, it all connects.
It does. So having only one copy of TBX1 instead of the normal 2, with a situation called haploinsufficiency, seems to be enough to disrupt the normal development of all these structures, leading to the spectrum of features we see.
Haploinsufficiency, half the dose isn't enough, okay? And there's another really surprising connection mentioned in the source. The high psychiatric risk linked to this specific deletion.
Yes, this is a critical and increasingly recognized aspect of 22q11.2 deletion syndrome. Individuals who have this deletion are at a dramatically increased risk for developing serious psychiatric illnesses later in life.
How high is the risk?
The source estimates that schizophrenia develops in approximately 25% of affected adults.
25%. Compared to what, about 1% in the general population?
Exactly. It's a massive increase in risk. And looking at it the other way, studies have found this specific deletion in maybe 2% to 3% of individuals who present with childhood-onset schizophrenia.
So it's a major genetic risk factor for psychosis. That link between a structural chromosome defect and psychiatric illness is really striking.
It is. It underscores how complex the brain's development is and how disruption of even a single region containing multiple genes can have profound long-term consequences on brain function and mental health.
Okay, now let's revisit Down syndrome, but specifically through the lens of the translocation mechanism. We already established that about 95% of cases are the straightforward numerical type trisomy 21, 3 separate copies of chromosome 21, strongly linked to advancing maternal age.
That's the common picture, yes, resulting from an error in meiosis called non-disjunction, where the chromosomes fail to separate properly.
But we need to focus on that crucial other 4% or so of cases. These arise not from an extra whole chromosome 21, but from a structural abnormality specifically, that Robertsonian translocation we discussed earlier.
Right. In these cases, the extra chromosome 21 material isn't floating free. It's physically attached, fused onto another acrocentric chromosome. Most commonly, it's fused to chromosome 14, so you have a dir1421 translocation chromosome.
Therefore, derivative.
Derivative, yes, meaning an abnormal chromosome derived from parts of 14 and 21.
Okay, so a parent, often the mother, is a balanced carrier. What does her karyotype look like?
The balanced carrier actually has only 45 chromosomes in total.
45, not 40.
Correct. Because one chromosome is the large, fused dir1421 chromosome. She'll also have one normal chromosome 14 and one normal chromosome 21.
Ah, I see. So if you count the chromosome 21 material, she has the equivalent of 2 copies, one normal 21 plus the 21 part fused to the 14, and the equivalent of 2 copies of chromosome 14 material, one normal 14 plus the 14 part in the fusion. So her gene dosage is correct.
Exactly. She has all the necessary genetic information, just packaged differently. That's why she is phenotypically normal.
But the problem arises when she makes eggs.
Precisely. During meiosis, when she's forming her eggs, the chromosomes segregate. There's a chance that an egg will inherit the fused in 21 chromosome plus her normal separate chromosome 21.
Okay, so that egg gets the fused 1421 and the normal 21.
Right. If that egg is then fertilized by a normal sperm, carrying one normal 14 and one normal 21, the resulting zygote will have one normal 14 from the father, one normal 14 from the mother within the fused chromosome, one normal 21 from the father, and 2 copies of chromosome 21 material from the mother, one normal 21 plus the one fused to the 14.
Making 3 effective copies of chromosome 21 material in total.
Exactly. The required triple gene dosage for chromosome 21 is achieved, leading to Down syndrome. But the key difference is the cause.
It's inherited from a balanced carrier parent.
Yes, which means this form of Down syndrome can be familial, it can run in families, and importantly, the risk is not related to the mother's age, unlike the much more common non-disjunction type.
That makes genetic counseling critical for these families, right? To understand the risk for future pregnancies.
Absolutely essential. Identifying a Robertsonian translocation carrier allows for accurate risk assessment, which can be quite high, and discussion of reproductive options. It's a completely different risk profile than age-related non-disjunction.
Okay. Finally, let's connect these structural aberration mechanics to the sex chromosome disorders. You mentioned earlier that imbalances involving sex chromosomes, X and Y, are often better tolerated than imbalances involving autosomes, chromosomes 122.
Yes. If we connect this to the bigger picture, it helps explain why conditions like Turner syndrome 45X or Klinefelter syndrome 47-7XXY are viable, whereas having an extra chromosome 1 or 2 or missing one is almost universally lethal very early in development.
Why is that?
There are two main reasons. First, the Y chromosome actually carries relatively few genes compared to the X or the autosomes. Most of its genes are related to male sex determination and sperm production. So having an extra Y, XYY, or missing it, leading to 45X if the sperm lacked a sex chromosome, has less impact on overall essential gene dosage.
Okay, the Y is less critical for general survival. What's the second reason?
The second and really major reason, particularly regarding the X chromosome, is the phenomenon of X inactivation, also known as the Lyon hypothesis, after Mary Lyon, who proposed it.
Right, X inactivation. Let's make sure we unpack that properly because it's so fundamental here. We're talking about the cell essentially deciding to shut down most of the genes on one of the X chromosomes in every single female cell, right?
That's the core idea, yes. Early in female embryonic development, in each individual cell, one of the two X chromosomes is randomly chosen to become inactivated.
Randomly chosen. So some cells inactivate the X inherited from the mother, others inactivate the X from the father.
Exactly. It's generally a random choice in each cell lineage. And once that choice is made in a cell, all of its descendants will keep the same X chromosome inactivated. So females are effectively mosaics, with patches of cells expressing the genes from the maternal X and patches expressing genes from the maternal X.
And why does the cell do this? What's the purpose?
The purpose is dosage compensation. It's to ensure that both males, who have only one XXY, and females, who have two XX, end up with roughly the same functional dose of X-linked gene products. By shutting down one X in females, it equalizes the expression level of most X-linked genes between the sexes.
Okay. And the inactivated X, what happens to it visually?
The inactivated X chromosome undergoes a dramatic change. It becomes highly condensed, tightly packed into a structure that's visible under the microscope in interphase cells known as a Barr body. It's transcriptionally silent for the most part.
So how does this X inactivation mechanism help mitigate the damage from structural abnormalities on the X chromosome?
It provides a crucial buffer. If a female has, say, a deletion on one of her X chromosomes or some other structural fault, the random inactivation means that in roughly half her cells, the normal X chromosome will remain active.
Ah, so she can often rely on the healthy copy in many of her cells.
Exactly. The normal X can carry the load, compensating for the damaged one in a significant portion of her tissues. This often lessens the severity of the clinical phenotype compared to what you'd see with a similar-sized defect on an autosome, where there's no such compensation mechanism.
But it's not a perfect fix, right? Structural faults on the X still cause major problems, especially in conditions like Turner syndrome, which is typically monosomy X or 45X.
Correct. X inactivation helps, but it's not a complete shield. And while the classic form of Turner syndrome is indeed 45, X meaning only one X chromosome is present, the source material highlights that a significant minority of cases, maybe around 14% or so, involve structural variants of the X chromosome.
And all these structural variants still lead to Turner syndrome.
They lead to features consistent with Turner syndrome because they all result in partial monosomy of the X chromosome. Some part of the X chromosome's genetic material is missing or non-functional.
Okay. And you mentioned earlier that the isochromosome is the most frequent structural cause here.
Yes, let's revisit that. The isochromosome of the long arm, IXQ10, is the most common structural abnormality found in Turner syndrome patients who don't have the classic 45X karyotype.
IXQ10, meaning two long arms, no short arm.
Exactly. The individual has effectively lost all the genes located on the short arm, the P arm, of one X chromosome. This results in partial monosomy for those XP genes, and that loss is sufficient to cause the features of Turner syndrome.
Are there other structural causes besides the isochromosome?
Yes. Other causes include specific deletions of parts of the short arm, written as delXP, or deletions of parts of the long arm, delXK, or even ring chromosomes formed from the XRX. All of these result in the loss of some X chromosome material.
Okay. And the molecular link for that really signature feature of Turner syndrome, the short stature, is very revealing, isn't it? It shows that X inactivation isn't actually complete. Some genes escape it.
This is a perfect example of dosage sensitivity and the limits of X inactivation. Short stature is an almost universal feature of Turner syndrome, regardless of whether it's caused by 45X or a structural variant involving loss of the short arm.
And it's linked to a specific gene.
It's believed to be primarily caused by haploinsufficiency, having only one functional copy instead of two of the SHOX gene.
SHOX, where is that located?
SHOX is located right on the distal tip of the short arm of the X chromosome in band XP22.33. It's also present on the short arm of the Y chromosome.
Ah, so it's in a region shared by X and Y.
Yes, the pseudoautosomal region. And critically, the SHOX gene is one of the genes that normally escapes X inactivation. It remains active on both X chromosomes in females and on the active X and the Y in males.
So normal individuals need two active copies of SHOX for normal height.
That seems to be the case. Two active copies are needed for proper skeletal growth. So an individual with Turner syndrome, whether they are 45X, only one X total, or they have a structural deletion involving XP, like the IXQ10 isochromosome, missing the short arm, ends up with only one functional copy of the SHOX gene.
Haploinsufficiency again.
Haploinsufficiency for SHOX. And this lack of the second dose directly impairs skeletal growth, leading to the characteristic short stature. It typically demonstrates how the specific structural location of a fault, combined with knowledge of which genes escape inactivation, dictates the clinical outcome.
Okay, so we've firmly established that routine banding, while foundational, has that major resolution limit. It's going to miss anything smaller than, say, 2 million base pairs.
Right. It's great for large-scale architecture, not fine details.
So the introduction of modern molecular diagnostics, really, as the phrase goes, changed the game for detecting these more subtle structural abnormalities, allowing us to find those cryptic microdeletions and microduplications.
Absolutely. And the prime example, the technique that truly revolutionized the field back in the 1990s, was fish fluorescence in situ hybridization.
FS.
Offered resolution far beyond standard banding. It could pinpoint deletions or duplications down to the kilobase level, which is absolutely essential for diagnosing conditions caused by microdeletions, like the 22q11.2 syndrome we just discussed, or others like Prader-Willi syndrome or Angelman syndrome, which are caused by deletions on chromosome 15.
Okay, FIS. How does it actually work? For our listeners who might not be familiar with visualizing DNA like this, how do you make it fluorescent?
It's actually a very elegant technique. You start by creating specific DNA tropes. These are short stretches of single-stranded DNA that are designed to bind or hybridize only to a very specific complementary sequence of DNA on a particular chromosome.
So you design a probe for the exact spot you're interested in.
Exactly. And crucially, these probes are labeled with fluorescent molecules that glow brightly under a special microscope when exposed to certain wavelengths of light. You can have probes that glow green, red, orange, blue.
Okay.
So for diagnosing the 22q11.2 deletion, for instance, a cytogenetics lab might use a cocktail of probes. They typically include a control probe, maybe one that binds somewhere else on chromosome 22 and glows green, just to confirm that chromosome 22 is actually present in the cell.
Right, a sanity check.
And then they'd include a test probe, specific for the 22q11.2 region they suspect is missing, and maybe label that one red.
Okay, green control, red test probe for the potentially deleted region.
Then you apply these probes to the patient's cells on a microscope slide, often the same metaphase spreads used for karyotyping. But FIH can also work on interphase cells, which is a big advantage. The probes find a bind to their target sequences on the chromosomes.
And then you look under the fluorescent microscope.
And the visual evidence is usually very clear. You look at the chromosome 22s. If the patient is normal, both copies of chromosome 22 will show both a green signal from the control probe and a red signal from the 22q11.2 probe.
Makes sense. Both present.
But if the deletion is present, one copy of chromosome 22 will look normal, showing both green and red signals. But the other copy of chromosome 22, the one with the deletion, will only show the green signal. The red signal will be missing because its target DNA sequence isn't there.
The absence of the red signal confirms the deletion. Very direct.
Extremely direct and visually convincing. FIH was a huge leap forward in resolution and diagnostic power for these kinds of structural changes.
Okay, now let's fast forward a bit to what might be the major consumer-facing advancement in the molecular diagnosis of chromosomal disorders, something many listeners might have encountered during pregnancy. Non-invasive prenatal screening, or NIPS, sometimes called NIPT for testing.
NEPS has been another major technological leap, yes, especially because it largely sidesteps the need for invasive procedures like amniocentesis or chorionic villus sampling, CVS, for initial screening.
Right, just uses a maternal blood draw. How does that work? How do you find the baby's DNA in the mother's blood?
It relies on the discovery that small fragments of DNA from the placenta, which is genetically representative of the fetus, naturally break off and circulate in the mother's bloodstream. This is called cell-free fetal DNA, or cfDNA.
Cell-free DNA, just floating around.
Yes, mixed in with the mother's own cell-free DNA, which makes up the vast majority of it. The fetal fraction, the percentage of cfDNA that comes from the fetus, is usually relatively small, maybe somewhere between 5% and 15% of the total cfDNA, depending on the stage of pregnancy.
Okay, only 5–15% is fetal. How can they reliably determine the fetal chromosome count from such a small fraction?
It works by sheer computational power and statistics using next-generation sequencing, or NGS.
NGS, okay.
They extract all the cfDNA, both maternal and fetal, from the mother's blood sample. Then they sequence millions upon millions of these short DNA fragments. They don't sequence the whole genome, just enough fragments to count.
They count the fragments.
Essentially, yes. Using computer algorithms, they map each sequence fragment back to its chromosome of origin. So they count how many fragments map to chromosome 21, how many to chromosome 18, how many to chromosome 13, and so on for all the chromosomes.
And then they look for imbalances.
Exactly. If the fetus has a normal chromosome count, you'd expect the number of fragments mapping to each chromosome to be proportional to the size of that chromosome, with slight variations. But if the fetus has, say, trisomy 21, Down syndrome, there will be a small but statistically significant increase in the proportion of DNA fragments mapping specifically to chromosome 21 compared to the reference chromosomes.
Because there's an extra chromosome 21 contributing its DNA fragments to the mix.
Precisely. The algorithms can detect this subtle dosage imbalance with very high accuracy for the common trisomies, like trisomy 21, trisomy 18, and trisomy 13, and also for sex chromosome aneuploidies.
It's amazing technology. However, the source material is careful to add a caveat. NEPS is primarily a powerful screening tool, not necessarily a definitive diagnostic test, especially when it comes to complex structural changes.
That is the absolutely critical takeaway regarding NEPS and structural abnormalities. NEPS is fantastic at screening for aneuploidy, detecting extra or missing whole chromosomes. It's much less reliable, currently, for detecting smaller structural changes like microdeletions, microduplications, or balanced translocations.
Why is that?
Well, detecting a balanced translocation with NEPS is basically impossible because there's no change in the overall amount of DNA, just rearrangement. For microdeletions or duplications, while some NEPS panels are starting to screen for common ones like 22q11.2, the detection rates and accuracy are generally lower than for whole chromosome aneuploidy. The signal from a small missing piece within that already small fetal DNA fraction is just harder to reliably distinguish from background noise.
So, if NEPS comes back positive or high risk, confirmation is still needed.
Yes, conventional karyotyping, often performed via amniocentesis or CVS, is still usually required for definitive diagnostic confirmation following a high-risk NEPS result.
And the karyotype gives you the full picture.
The karyotype, often supported by FASH or maybe chromosomal microarray analysis, which is another powerful tool for detecting smaller gains and losses, gives you the complete structural picture. It can confirm the aneuploidy, but importantly, it can also identify the cause. Is it simple non-disjunction, or is it due to an unbalanced translocation? It can pinpoint specific breakpoints, detect balanced rearrangements that MIAPAS would miss entirely, and provide much more detailed information needed for accurate genetic counseling, especially regarding risks for future pregnancies. NEPS is a phenomenal screener, but it doesn't replace the detailed map provided by cytogenetics for complex cases. #conclusion.
Well, this deep dive has certainly shown us that the physical integrity, the actual structure of our chromosomes, is just as crucial and maybe even more fragile in some ways than simply having the correct number.
Absolutely.
We've really covered the whole spectrum of these structural aberrations today, from things like balanced inversions or translocations that might be phenotypically silent in the person carrying them, all the way to large deletions, like that 22q11.2 example, that cause complex multi-system syndromes with really significant health consequences.
And understanding that precise address, the cytogenetic nomenclature like XP21.2 or 16p11.2, p13.1, we saw how fundamental that is for diagnosis, for predicting risk, for managing these conditions effectively.
Yeah. It's amazing how one single break or a faulty fusion after a break can potentially lead to lifetime consequences for an individual or fundamentally determine their ability to have healthy children.
It really underscores the importance of accurate DNA repair and faithful chromosome segregation during cell division.
So, stepping back, what does all this mean for the bigger picture of human genetics? What's the main takeaway here?
I think the key takeaway is that structural integrity is absolutely paramount. And the cellular mechanisms we rely on to maintain that integrity, like repairing DNA breaks, are powerful but inherently imperfect, prone to error. We saw that while carriers of balanced rearrangements like translocations or inversions may appear perfectly normal themselves, they often carry this high inheritable risk of producing unbalanced gametes, leading to problems for the next generation. That highlights the crucial difference between having a balanced genome in your body cells versus transmitting a balanced set to your offspring.
Right.
And the disorders that we actually recognize clinically, the translocation Down syndrome cases, the Turner syndrome structural variants, the 22q11.2 deletions, these are really just the survivors. They represent only that small fraction of structural errors that are actually compatible with survival past the earliest stages of development.
Which brings us back to that staggering statistic you mentioned earlier.
Yes, the source material estimates that something like 7.5% of all human conceptions might have some kind of chromosomal abnormality, numerical or structural. The vast, vast majority of these are simply incompatible with implantation or continued development, resulting in very early pregnancy loss, often before it's even recognized.
7.5%, that's huge.
It is. And it really leads to a final, maybe provocative thought for our listeners to consider.
Okay, let's hear it.
Considering the inherent fragility of chromosome structure that we've discussed and the surprisingly high frequency of errors that occur, particularly during the complex process of meiosis, when structural rearrangements can cause such havoc, how truly robust is this instruction manual of the human genome? And perhaps more interestingly, what are the immediate ongoing evolutionary pressures that are driving the development and maintenance of complex biological mechanisms like X inactivation, or perhaps others we don't fully understand yet, that seem designed specifically to help us cope with this high background rate of inevitable structural errors? It almost seems like our survival as a species is less about achieving perfect genetic fidelity, and maybe more about having evolved efficient, if sometimes imperfect, ways to buffer ourselves against or mitigate the effects of the constant structural challenges our chromosomes face.
Wow. So evolutionarily, maybe we're built not just to be correct, but to tolerate being incorrect up to a certain point. That's a truly profound thought. It shows the genome isn't just a static blueprint, but a dynamic system constantly battling its own potential for structural chaos. Thank you so much for guiding us through this really complex but fascinating deep dive today.
My pleasure. It's a critical area of genetics.